So in the previous lecture, you were introduced to this idea of internal and external environmental context contributing to the actions or effects of the brain and behavior responding to the environment. For this lecture, we're going to be spending a good amount of time discussing a proximal cause of behavior and brain function, and that's genetics. So Darwin, in his observations, recognized that animals that were best suited for their environment were able to pass on their genes to the next generation. So in this particular example, we have the rock pocket mouse, and they come in variations of two colors, brown and black, and these specific colors have survival value given the environmental context in which these mice live. So colors that blend into the environment, as shown in these two panels up here, the brown with the light beige background and the black with the lava background, are better hidden from possible predators and can therefore be protected from situations where they could die. In contrast, animals that aren't as well hidden, so the black against the beige and the brown against the black, are much more prominent in the fact that they stand out, and so they're more likely to die off and therefore not pass on their genes to their offspring. And so Darwin hypothesized, right? Darwin, remember him? So based on his observations in the field and under the microscope, he proposed or hypothesized that the biological mechanism of transmission of these characteristics was a unit that he wasn't aware of at the time because the technology didn't exist, but he called it a gemmule. What's interesting is that around the same time, this guy over here, an Austrian monk named Gregor Mendel, was trying to determine the unit of inheritance, but what he did was he was studying pea plants to figure out how characteristics are inherited. So he studied pea plants, and he figured out that some traits were suppressed and others were expressed in subsequent generations, leading to situations where variable heritable differences existed in shape and color. So he figured out that there were genes, he didn't know what these were at the time, but something within the cell contribute to the phenotype, which is the expression of a particular trait. So these observable traits and characteristics he called a phenotype. And so let's talk a little bit about the basics of inheritance before we talk about the way in which Gregor Mendel figured out how genes are passed on. So most of us have two copies of a gene. When we inherit genes from our parents, we usually get one copy from our mother and one from our father. Each of these copies is a version of the gene, so they aren't exact, and these versions are known as alleles. Okay? Some alleles, some versions, are dominant and others are recessive. When you have one copy of the dominant gene, regardless of which parent you get it from, that particular phenotype will show up. So you can have one dominant phenotype and one recessive phenotype, and the dominant phenotype will show up. So whenever you have one copy of the dominant allele, the dominant phenotype will show up. When you have two copies, the recessive phenotype will only show up if both alleles, both copies of the same gene, are recessive. That's the only way that you'll have a recessive phenotype showing up. So when you have two copies of the same gene, it's known to be homozygous. When you have two different copies of the gene, so one allele is dominant and the other recessive, that's known as heterozygous. So Mendel looked at two genes, one for color and one for texture. At the time, he didn't know that they were two different genes until he did his experiments, but he decided, I'm going to take a look at the color and texture because they seem to be varied in this pea plants. So he looked at the color and texture of plants. He found that they were either smooth, which is denoted by the big S, or wrinkled, which is denoted by the little s. So any trait that's recessive or any gene or allele that contributes to a recessive phenotype is denoted with a smaller letter of the alphabet, where the dominant allele is usually denoted with the capital letter. And so same thing here, color is either yellow, which happens to be dominant, or green. So a pea plant that is yellow and smooth gets this genetic makeup. So it has 2 copies of the dominant allele for both texture as well as color. Just to reuse the terms we talked about before, this particular pea plant is homozygous for color and texture. In this instance, this particular pea plant is homozygous for color and texture as well, but it happens to have 2 copies of the recessive allele for texture and 2 copies of the recessive allele for color. So it happens to be wrinkled and green. Okay, now that you have the basics of the terminology, let's apply it to what Mendel actually did. So here's what he did. He took smooth yellow plants and mated them with green wrinkle plants. And what he found in the offspring of these pea plants, which is known as the F1 generation, because they're first generation offspring, is that all of them were yellow and smooth. At the time, he didn't have this information about the genes, but we now know that each plant, each animal, gets 1 copy from each parent. So this particular pea plant got a big S from this parent here, and this little s from parent number 2. Same thing goes for the yellow color. Okay, so this particular pea plant, the F1 pea plant, is actually heterozygous for color and texture. And then what he did was he decided, I'm going to mate the offspring together. So he mated these heterozygous offspring of the F1 generation, and the probability of what he got looks like this. Okay, so each one of these boxes refers to the different kinds of plants or different kinds of peas that this particular mating structure can get, right? Some of them have dominant traits, some of them have recessive traits. They're dominant for color but recessive for wrinkliness, as seen here. Some of them are dominant for smoothness but recessive for color, as seen here. So different combinations of alleles and genes result in different phenotypes, as shown here by this box. So this represents the 2nd generation, or the F2 generation. But you guys don't need to worry about any of this information, but you should understand the ways in which genes are transmitted, and it has to do with whether you have a dominant allele or a recessive allele for a particular gene. And what's incredible is that Gregor Mendel, figured out all of this without any fancy technology. He correctly deduced the mode and rules of inheritance that still hold true today, 150 years later, which to me is absolutely incredible. So how do genes translate to phenotypes? It has to do with this. So within the nucleus of a cell, right, shown here in red, there are 23 pairs of chromosomes. These are the chromosomes that are tightly packed within your nucleus. These chromosomes happen to be tightly wound around protein balls that look like spools and that can be unwrapped or wrapped tightly. This is known as chromatin. Okay, chromosomes are made up of chromatin, and chromatin is actually made up of DNA, which is tightly wrapped around these protein spools that we will talk about some other time. That isn't important for this slide right now. But DNA is a helical structure containing the instructions for life. That's what you see right here. And we have like yards and yards and yards of DNA tightly wrapped into every single nucleus of our cell. Isn't that incredible? So the code, the instruction for all life, exists in the form of what are known as nucleotides. So within this helical ladder, the rungs of the ladder come together in pairs, and the order of these nucleotides represents the recipe for protein. So literally all DNA is recipes for every single protein that our body makes. And depending on the cell type of your body, different proteins are needed. And so different genes are turned on or turned off as the cell needs it. So every single one of your cells contains all possible genes. They're identical, but which ones get expressed depends on the type of cell that is expressing that protein. So you might wonder, like, who figured all of this out? How did it work? This work of deciphering the structure of the DNA was published by Watson and Crick. They received the Nobel Prize for decoding the helical structure of DNA, but, and it's a big but, if it wasn't for the work that they stole from Rosalind Franklin, shown here on the right, they would have never figured out. So Rosalind Franklin was an X-ray crystallographer, and she was taking X-rays of DNA, trying to figure out the structure. And one of her colleagues showed her X-rays to Watson and Crick without her permission, and based on that information, Watson and Crick figured out the structure of the DNA. So the credit goes to all three of these scientists, but unfortunately Franklin died from ovarian cancer at the age of 37, likely from all of the radiation she was exposed to during her research. So the data, which was the data that Watson and Crick used to figure out the helical structure of the DNA. So we have to kind of rewrite history. Watson and Crick won the Nobel Prize for this work, but Rosalind Franklin's work has to be noted because without her X-ray data, they would never have figured it out. Okay, so since Watson and Crick figured out the structure of the DNA, a whole bunch of other scientists over many decades figured out how this code was used to make every possible protein in your body. And that's what DNA is, as I mentioned. It's the recipe for how to make different proteins. The simplest recipe ever is to make the variety of proteins that do so many different things. Proteins that we'll be learning about in the coming weeks. So the way in which proteins are made is through a process of transcription and translation. So you know that DNA resides in the nucleus and it never leaves the nucleus, but all of the machine to make proteins resides outside of the nucleus. So you need kind of a middleman to bring this information from the nucleus to the outside, and that's what the mRNA is. So the messenger RNA is used to kind of relay the message to the outside of the nucleus through the process, and making mRNA is known as transcription. You're using DNA to copy the code into mRNA. Then the machinery, so the mRNA travels to the outside of the nucleus, and the protein is made using the mRNA. So there's a specific machine, called ribosomes that we'll talk about later, which is used to make protein. But the process of making protein is known as translation. It's actually quite an incredible process that we won't have time to talk about, but this is essentially how it occurs. For those of you who are interested in learning more about this, because there's a huge field of neuroscience that studies molecular biology and applies it to brain function, this is kind of like what they're studying. They're trying to understand how mutations in your genetic code can lead to dysfunctional protein and cause diseases like Huntington's disease, for example. So makes you wonder, right? So unlike some diseases, as I just mentioned, like Huntington's disease or Tay-Sachs disease, that typically involve just one gene, most functions in the brain or disorders of the brain result from alterations in the combinations of genes and proteins. So together, a whole bunch of altered genes work together to result in a disease. Additionally, we have no idea, additionally, we now know that environment, right, what you're exposed to on a daily basis, can also dictate which genes are activated and which genes are not. These changes in gene expression can actually be inherited, and there's a huge field of study that's exploring this phenomenon in different subjects, and the field is known as epigenetics. So just to give you an idea of what I mean by epigenetics, I wanted to tell you about the Dutch winter famine that happened in 1944. So around the time of World War II, when the Dutch were under Nazi control, they they shut off all food to the Dutch people. And people in cities really suffered the most because they had no way of getting food. Those that lived out in, rural farmland area could grow their own food, but those in the cities really relied on the food coming into the city. But during the winter, there was absolutely no food because the Nazis cut off that supply. And so a lot of people died as a result of starvation during this winter. But interestingly enough, there were some women who were pregnant during this time, during the famine, and as a result, they had kids, and those kids grew up. But what was interesting is that researchers that followed these kids, they found that when these kids became adults, they ended up gaining a few more pounds than the average. At middle age, they had higher levels of bad cholesterol, triglycerides, and LDLs, and they also experienced higher rates of such conditions as diabetes, obesity, and schizophrenia. How is it possible that kids who might have grown, had the same condition as other kids after they were born, were more susceptible to these kinds of diseases? It turns out it directly correlated with when their mothers were pregnant with them during the famine. So what happened was that as a result of that famine, the genes that the the kids had in the environment were altered so that when they became adults, the genes behaved differently in their expression than somebody whose mother didn't undergo the famine during pregnancy. So epigenetics, which is changes being made to the outside of the gene, not the actual code itself, but to the outside of the gene, these genes or these changes can affect what genes are expressed and what are not. And the environment has a direct impact on the expression of these genes. And so the kinds of changes that can be made to the genetic code or the expression are things like this. A methyl group may be added, a chemical is added to the DNA, or to the proteins that the DNA is wrapped around, ultimately controlling whether the gene gets expressed or not. So during the famine, when the moms were pregnant with these kids, the environment, the lack of food, changed some of the genes of the kids, who were fetuses at the time, ultimately resulting in them being more susceptible to developing things like obesity and schizophrenia. And so in this way, environment can impact the expression of genes and ultimately affect things like whether you're more susceptible to developing a disorder like schizophrenia. All right, we're going to shift gears a little bit and talk about the different ways in which scientists can use different techniques to study the human brain. All right, so when you look at this horrible hunk of tissue, it's really not that impressive. It's hard to imagine that this chunk of brown contains all that exists in who we are as people, our consciousness, our thoughts, our feelings, our experiences, right? This hunk of tissue contains all of that and allows us to function as normal human individuals. Like things that we take for granted is entirely the result of the actions of our brain. That's pretty incredible. So scientists use different ways to better understand how the brain controls such complex functions. One of them is through the use of case studies. So this guy over here, he was a construction worker that had a nail gun, amazingly, shoot off a nail that went into his head. He didn't realize the nail was embedded into his head and continued working through the day. But later on in the evening, he started getting nauseous, had headaches, started throwing up. And so he went to the doctor and they found a nail embedded in his skull. The doctors were amazed that he survived and he was functioning normally. And so they took the nail out successfully and were able to He was able to go back to his daily life. But case studies such as these, right, because it's not ethical for us to shoot a nail into a person's head or destroy parts of their brain, when it happens accidentally, we can better understand the ways in which certain brain areas control functions. If those brain areas get damaged with a stroke, for example, or with some sort of injury, we can better understand how that brain controls particular behaviors that might change following the stroke or the injury. Another way in which we can study the brain is through the use of electroencephalograms, or EEGs. These are electrodes that are applied to the scalp of the brain, and they end up detecting electrical activity on the surface of the brain. What's really interesting is that researchers can use this study to see how the brain responds to specific stimuli in the environment. So these are known as event-related potentials. So in the brain, when you present stimulus of some sort, whether it's visual or auditory, you can watch and see how it changes the electrical activity of the brain. Here's an example of an event-related potential following the presentation of a stimulus right here. After a certain amount of time, after about 400 milliseconds, you see an increase in activity within the brain, representing the brain's response to that particular stimulus that was presented. This particular technique can also be used for sleep research, which we'll be discussing later in the semester. You can also use ERPs to kind of see how people respond. In this particular example, they noticed that people with narcolepsy and Parkinson's disease responded differently to humor. So they created an experiment where they could actually measure the brain's response to humor. So they presented participants that were either healthy controls, had narcolepsy, which is a sleep disorder, or Parkinson's disease, which is a motor disorder, presented them with a cue, and then either provided a neutral feedback, which was just the image flipped over, or a humorous feedback, represented by the dog also waiting for its pat down. They measured the ERP in the brains of these participants, and what they noticed was that the healthy participants responded to the humor condition as well as the narcolepsy patients, but the Parkinson's patients, actually, their brains did not respond to the humorous cue or humorous feedback, indicating that Parkinson's disease changed their brains in some way that prevented their ability to recognize or process the humorous feedback. So in this way, we can use ERP or EEGs to better understand the ways in which the brain in living organisms and living human beings respond to environmental stimuli. If you want greater resolution, so if you want to see how individual neurons actually behave in response to stimuli, because remember the EEG is measuring huge populations of brain cells, you can do single cell or single unit recording from individual neurons using this really fine electrode here that is approaching this brain cell or neuron here. If you're interested, for example, in studying the activity of what alcohol does to the brain, you can use something called positron emission tomography, or PET. What they do is they administer radioactive substances that don't last very long, and then a computer measures or scans the brain for radioactivity, and you can see what brain areas are most active. So in this situation, they gave healthy controls either placebo, shown here, or following alcohol, and then measured activity of the brain. So the redder areas of the brain represent more active, and not surprisingly, following alcohol consumption, either of 0.5 grams per kilogram or 0.25 grams per kilogram, the activity level in the brain decreased substantially. You can also use MRI, magnetic resonance imaging, or DTI, diffuse tensor imaging. Both of these techniques will allow you to see different things within the living human brain. The MRI will allow you to see all parts of the brain and make spaces appear black. DTI, diffuse tensor imaging, allows you to see white matter tracks. They appear brighter than gray matter tracks. So white matter tracks are those parts of the brain that carry information from one side to the other. And what they're doing here is they're looking at a control healthy man as well as an alcoholic man, both the same subjects, either in an MRI or a DTI. And what you'll notice is that the spaces in the brain of the alcoholic man are much, much larger, which is indicative of long-term alcohol consumption. Furthermore, you'll notice that the white matter tracks are smaller and not as bright in the alcoholic patient compared to the control patient. So this represents the fact that long-term chronic use of alcohol can actually change the brain of people. In this particular type of technique, you can study the activity level of the brain on top of the brain structure itself. So this is called an fMRI, functional magnetic resonance imaging. And in this particular study, they compared alcoholics to controls to look at their activity level in the brain. And you'll notice that different areas of the brain are lit up in the alcoholic's brain that are not in the healthy control. Again, but then you have areas like the cingulate, which is an area deep within the brain that's active in the control, but not so much in the brains of alcoholics. So again, this represents the fact that structurally the brain changes with chronic alcohol consumption, but functionally as well, the brain works differently in somebody who's been consuming alcohol for many, many years compared to healthy controls. In those situations where we can't use human subjects because we are really trying to look deeper into brain function, we resort to animal models. And so rodent brains appear to have very similar structure and makeup as ours, and so they've been used for decades as the animal model to better understand how our brain controls various functions. The rat brain engages in more complex functions than the mouse brain. So if you're studying how rats make, or how the brain makes decisions, for example, you might opt to choose the rat instead. However, the mouse lends itself to more genetic manipulation. There are dozens of strains of mice that have been genetically altered to miss a particular gene. These are known as knockouts. And in this way, you can observe, using the mouse, the ways in which particular genes contribute to brain function and behavior. So if you're using an animal, one of the things you can do with an animal that you can't do with a human, unfortunately, is lesion or stimulate different areas of the brain. So there are many ways to determine function of brain regions. Ideally, you'd want to use multiple techniques to demonstrate the same thing. So one way in which you could determine if a particular brain region participates in a particular function is by destroying it or lesioning it. Okay, that means the same thing. You could use an apparatus that looks like this. This is known as stereotaxic surgery, where the animal goes into the apparatus and using an atlas shown here, precisely targets specific areas of the brain that you'd like to destroy. And what they typically do is they use high current electricity to destroy the brain area. So following recovery from surgery, scientists can then test the rat or the mouse for alterations in specific functions or behavior. And to verify that a particular brain area is involved in a specific behavior, you'd also want to verify it with other methods. So brain stimulation is another way to do this. Again, using stereotaxic surgery, where's my little pointer, shown here, you can stimulate the brain area instead of destroying it to see if the animal behaves with increases in a particular behavior that you thought that brain region controlled. And so your results from the lesion experiments will be more valid if you can verify it using the stimulation methods. So in this way, using multiple techniques, behavioral neuroscientists or biological psychologists can verify the function of a particular brain region. You can also examine specific behaviors. So, for example, if you have a mouse model for autism, you can put the mouse in this 3-chambered cage here and give the mouse the opportunity to be by itself, in this cage here, be it with nothing, or with another mouse. What they found is that animal models of autism, right, mice that have genetic alterations to represent autism, are less likely to choose social interaction, are less likely to interact with other mice, and are more willing to stay by themselves, which is one of the symptoms seen in individuals with autism. All of these studies in mice, unfortunately, in the laboratory, don't represent the conditions under which animals might encounter in the wild. So if you'd like to kind of see how an animal might behave in a natural setting, like not in a cage, you can do something called field research, right? So doing research in a laboratory is nothing like what an animal might encounter in the wild, so researchers can engage in field research. And so rather than manipulating the settings, researchers can just quietly observe animals in their natural settings and try to figure out the functions for these particular behaviors. So here's an example of a snub-nosed primate, snub-nosed monkey. What they found is they live in very, very cold areas, and they tend to have more fur and huddle together a lot, and it's likely that they huddle together a lot to conserve heat because they live in places that are very, very cold. So let's summarize this particular lecture. So we've learned that genes code for proteins, and these proteins ultimately influence a cell's function within circuits of the brain to give rise to complex behavior. So cells working together are known as circuits within the brain, and then they're the ones that contribute to the complex behavior we see in human beings. Transcription is the process of making mRNA, while translation is making protein using mRNA as the recipe. We've talked about epigenetics, where the environment influences the expression of particular genes. There are many different ways that we can study the human brain. In human beings, we can use things such as case studies, EEGs, positron emission tomography, DTI, MRI, or fMRI to better study it in living individuals. But in those conditions which requires manipulations of the brain in some way, we can use animals instead. So for single cell recordings, genetic manipulations, lesion and stimulation study, as well as behavioral testing and field research, we can use animals to better understand the human condition. So that's it for this lecture. Don't forget to submit the reflection for it.